A very good morning, students. Today we are going to study the last part of this 8th chapter, Microbes in Human Welfare, that is Fermentation Beverages. So far we have studied about the microbes that is used in household products, then microbes that is used in industrial products. One part that we have seen it, that is antibiotics and its production, as well as antibiotic resistance. And now we are going to study about the fermentation beverages, and this is also included under microbes in industrial products. And yesterday's class we have discussed about two different topics. One is microbial fuel cell. So how you can produce electricity, at the same time how can we treat the sewage water treatment by using the bacteria, microorganisms. And then we studied same microorganisms used for production of biogas or gobar gas. And today's topic is fermentation beverages. In this fermentation beverages, we are going to study how the microorganisms are used for production of beer, wine, whiskey, vodka, rum, all these extra products, that fermentation beverages. And regarding this, we have an industry which is running in Bangalore for the production of beer and wine. So those students who have done degree courses in this fermentation beverages will have a very good posting in those companies. Okay. So now we will study about the fermentation beverages. Microbes, especially yeast, is being used from time immemorial for the production of beverages. So we are using here yeast for production of wine, beer, whiskey, brandy and rum. So wine, one more question. Wine is the most among the oldest alcoholic beverages known, and is produced by fermentation of fruit juice by yeast. So how wine is produced? By fruit juice as well as yeast, two things. And very important, two more questions, zymology, very important. You can trust this question for your public exam. Zymology is an applied science that deals with two things: biochemical processes, fermentation and its practical uses. So this is what the zymology is. Next one, another important one more question, the Saccharomyces cerevisiae. It is already, I have told you, it's a brewer's yeast, a book pack question. So Saccharomyces cerevisiae is commonly called brewer's yeast. It's used for fermenting malted cereals, fruit juice, to produce various alcoholic beverages. Now wine and beer are produced, one more question, without distillation. So two things, wine, beer, they are just produced only by fermentation, no distillation is used. Whereas whiskey, brandy, rum, all these are obtained by two things, by fermentation as well as distillation. So they will ask you this as a one mark question. Clear? Right. So next one, we are going to study about how this wine is being produced. So the science and the study of wine and wine making is called oenology. So wine is made from a fermentation of grape juice. Fermentation, I already told you, it is getting one product is spoiled by using microorganism to gain the another product. So now here the grape juice is fermented by various strains of, that means types of, Saccharomyces cerevisiae into alcohol. Now there are two type of grape wine is there. One is red wine and white wine. This is a 2 mark question. Actually every paragraph you have to study. Any paragraph can be asked for your 5 mark question. But fermentation beverages together is a 5 mark question. Full paragraph. Okay, right. So red wine, and for red wine we are using black grapes. And they are when you use the black grapes, you are using the skin, and sometimes the stem also being used for this. And to opposite to this, we are using white wine. White wine is produced from juice of either white or red grapes without the skin and the stem. So this is the difference between red as well as the white wine. Understand, right? So next one, beer production. Now this beer production is produced from germinated, all these are one mark question. Okay, germinated barley, malt grain. This is also used by the two yeast that is called Saccharomyces Calbergenesis, as well as Saccharomyces cerevisiae. Next one is rum. Rum is also made from fermented sugarcane or molasses. Molasses is a waste product of the sugarcane, or directly from the sugarcane juice, and it is used by Saccharomyces cerevisiae. All these are important. Everywhere we will find the Saccharomyces cerevisiae. Next one is whiskey. Whiskey is a distilled alcohol only, and it is also made from fermented grain mash by Saccharomyces cerevisiae. Clear? So the next one is, in some part of South India, a traditional drink called padanir is obtained from fermented sap of palms as well as the coconut trees. Now, a common source is tapping of unopened splash of a coconut, and it is a refreshing drink which on boiling produces jaggery, or you can use some palm sugar. You boil along with this, so you will have this padanir. So when padanir is left undisturbed for few hours, it is getting fermented to form toddy. That will with the help of natural occurring yeast, and to form beverages that contain, one more question, 4% alcohol. So toddy will have 4% alcoholic. Then after 24 hours, toddy become unplated, as well as it is used by Saccharomyces cerevisiae and is the major production of ethanol. Then after 24 hours, now the toddy become unplatable, and it is used for the production of, one more question, vinegar. So toddy, if you keep it for 24 hours, it becomes vinegar. So first we have padanir, then toddy, and finally we get the vinegar. And next one is another important 2 mark question, industrial alcohol. So here you should understand certain subtopics here. First one, the production or the producer of ethanol. So here we use the major source for production of ethanol is Saccharomyces cerevisiae. Okay, so that is the first topic, that producer of ethanol. And second topic is uses. And today the ethanol is used for industrial products, laboratory and fuel purposes. So ethanol is referred to as industrial alcohol. 2 mark question. Okay. So now the third one is the microbacteria that is used for the ethanol production, so called Zygomonas mobilis and Sarcino verticuli. So these are the microorganisms that is used for, or bacteria used for ethanol production. Okay. And the fourth subtopic is the principal substrate for ethanol production. And today we have got certain principal substrate for ethanol production are the the principal substrate for the commercial production of industrial alcohol includes, all these are 1 mark question, molasses. Molasses is a waste product or end product of the sugarcane juice or corn, potatoes and wood waste. And next, finally, fifth one is the process of ethanol production. Now here the ethanol production starts by milling a feed of stock, followed by addition of dilute or fungal enzyme called amylase, and this is extracted from aspergillus to break down the starch into fermentable sugar. So we use an enzyme called fungal amylase, and this enzyme is obtained from aspergillus, and that is being used to break down the starch into fermentable sugar. And here we also included yeast, and yeast is added to convert the sugar to ethanol, which is then distilled obtained, and from that we can obtain the ethanol. So the ethanol which we obtained here is 96% in concentration. The two most common type of biofuel is used today are two things, very important. One is called the ethanol, another one is called the biodiesel. So both of them represent the first generation of biofuel technology. And next, 1 more question. Ethanol is often used as a fuel, mainly as a biofuel additive for gasoline. Clear? Right. So this is an important one. And next topic here, we are going to discuss about the microbes that is used in industrial product. So 3 subtopic we will be discussing about microbes used in industrial product. First one is the antibiotics, second one is fermentation beverages, and finally chemicals, enzyme and other bioactive molecules. Now here the microbes are not only used for commercial and industrial production of alcohol, but also used for the production of chemicals like organic acid as well as enzymes. An example of organic acid producers are, one mark question, Aspergillus niger for citric acid production. All these are one mark question, very important. At the same time 3 mark, 2 mark questions also be asked from this. Then Acetobacter aceti for acetic acid, Rhizopus oryza for fumaric acid, Clostridium butyriceum for butyric acid, then Lactobacillus for lactic acid. So all these are the microorganisms we use for production of certain organic acid and enzymes. And now here the yeast called Saccharomyces cerevisiae and the bacteria both are used for commercial production of enzymes. So here we have an enzyme called lipase enzyme or lipase. These enzymes are used in detergent formulations and are used for removing oil stains from the laundry. As well as we use some enzymes for purification of the bottled juice for clarifying these called pectinase, protease and cellulase. So these enzymes today used for bottled juices are clarified by this. Then the last one here, the rennet, that also can be used to separate milk into the solid curd for cheese making. So these are all the enzymes. So first part we studied about the organic acid, and this is second part is for the enzymes. And next one, streptokinases is another 2 mark question, very important, very important. Trust this question, called clot buster. Here we use an a microorganism, an enzyme product which is produced here. Streptokinases is an enzyme. Always when it ends with ase, it is called enzymes. So streptokinases, they are produced from the bacterium called Streptococcus bacteria. Today this bacteria is genetically engineered streptococci, and today that are used for clot busters. Clot busters means if any clot is there in the blood vessel, so that clot can be removed by the patient, so clot is removed from the blood vessel by using this enzyme called streptokinases. If you give this enzyme, the clot is removed. So this kind of enzyme is today obtained from a bacteria called Streptococcus bacteria. Okay, so all these are very important, and this is for treating for all myocardial infarctions, that is closely related to some heart failure, all this. Okay, right. So now today we use these kind of microorganisms and their products for valuable products, even in many pharmaceutical industries as well as some commercial industries and all. Today we are using these microorganisms. So those students who have completed microbiology in degree courses, they will be appointed for all these courses later on, and they will get a future very good job opportunities, job carries will be there for those students who have completed this microbiological science. Clear? Right. So that is important. And next one here, we will study about certain some antibiotics as well as some microorganisms related to the chemicals. Now here, next one is cyclosporin A. Now the cyclosporin A is also called as immunosorbitant, that is used in organ transplantation. Now here, immunosorbitant means if an organ is getting transplanted from one person to another, sometimes there may be some allergic reaction will be there. So in that case, we give some drugs for suppressing the antibody production in our body. So if antibody production is lowered, so the organ which you donate can be accepted by the donor, can be accepted by the recipient. Okay, so now here we use cyclosporin A for organ transplantation, and this cyclosporin A is obtained from the fungus called Trichoderma polysporum. 1 mark question. And it is also used for its anti-inflammatory number 1, antifungal number 2, and anti-parasitic properties. Clear? So they will ask you 1 mark question, which one is used as an immunosuppressant, and which one is used as a anti-inflammatory, antifungal, anti-parasitic properties. All this are called cyclosporin A, and is obtained from Trichoderma polysporum. Clear? Right. And next one is a strain. The strain that is produced from a yeast called Monacis purpurus. and that has been used to lower the blood cholesterol level. Very important. It will lowers the blood cholesterol level, as well as it will act as a competitive inhibiting the enzyme responsible for synthesis of cholesterol. Okay, finally we are discussing about the recombinant human insulin today, and that has been produced by using two things, E. coli as well as Saccharomyces cerevisiae, for therapeutic use in human. Clear? Right. So far we have studied about the microbial products, everything. Clear? Right. So this lesson you have to give very importance to certain topics, as well as very importance to all the microorganism, their product and the uses. Three things you have to study: microorganism, they are where from where they are produced, and for what they are used. So these three things are important. Simultaneously you should study all the paragraphs. There is no this and that. Every paragraph is important. So sequentially you study. First you start studying about the antibiotics together, fully antibiotics to resistance. Second, you start studying the fermentation beverages up to above paragraph chemicals. Then start studying all the chemicals. Then go for MFC, then gobar gas or biogas. And finally we have to study about the bioremediation. Now the next topic that finally we will study about the bioremediation. What is bioremediation? Is using microorganisms to cure the or to remove the pollutions in the environment. So that is what the bioremediation are. Remediation for purifying the environment pollution by using certain microorganisms. Now the use of naturally occurring or genetically engineering microorganisms to reduce or to degrade pollutants is called as bioremediation. Very important 5-mark question, this fully. Okay, book back question, please study. Bioremediation is a less expensive and more suitable than other remedies available. It is grouped into two things. One is called in situ, another one is called ex situ. In situ bioremediation means treatment or curing the contaminated soil and water inside our area, that is called in situ. Ex situ means treating or removing the contaminated soil and water that is from the site, that is from not within our area, from other areas. So that is what the difference between in situ and ex situ bioremediation is. Now in this we are going to study two things, two type of microorganisms. One is aerobic microorganisms and another one is anaerobic microorganisms. So now first we will study about the microorganism involved in bioremediation, that is aerobic microorganisms. Here in this aerobic microorganism, they degrade the pollutant in the presence of oxygen, and they mainly degrade two things. One is called the pesticides, another one is called hydrocarbon, which are highly pollutant, highly poisonous. Now 1-mark question. The Pseudomonas putida is a genetically modified microorganism, that is otherwise in short we will call it as GEM. It is produced by or obtained produced by Anand Mohan Chakrabarty. He is an Indian born scientist. Obtained patent for this recombinant bacterial strain. So this strain is called superbug, already I have discussed with you, and it is a multiplasmid hydrocarbon degrading bacteria, 1-mark question, which can digest the hydrocarbons in the oil spill. If any oil tankers are getting accident and oil is spilled in the ocean, these oils are removed by this kind of a superbug. They spray this bacteria and come, so this bacteria will eat up all the oil spills. Clear? Right? That is number one. So that is the thing that is I have given it, the process of bioremediation. Later on we will discuss about this diagrammatic part. So now we will go for this interior part of it. Now the second microorganism is called as Nitrosomans europea. It is also called, it is also capable of degrading, 1-mark question, benzene and a variety of halogenated organic component including trichloroethylene and vinyl chloride. Another one is a book back question, Ideonella sacchiensis. It is currently tried for recycling of PET plastic. The bacteria use a PETase, that is PETase, and MHETase enzyme to break down a PET plastic into terpenoic acid as well as ethylene glycerol. So all these are the important 1-mark question when you study about this aerobic microorganisms. And now we will talk about anaerobic microorganisms. An anaerobic microorganism degrade the pollutant in the absence of oxygen. And here we have Dechloromonas aromatica. It has the ability to degrade benzene. So there also we studied benzene, here also benzene, but the difference is there we studied about the aerobic bacteria, and here we are studying about the anaerobic bacteria. And oxidize toluene and xylene. Clear? So that is one. Then Panirochete chrysosporium is another anaerobic fungal, one mark question, not bacteria, fungus. It exhibits strong potential for bioremediation of pesticides, then polyaromatic hydrocarbons, then dyes, then other one is trinitrotoluenes, then cyanides, carbon tetrachloride, all these are. And here finally we will discuss about the another important one mark question, that is the Dechlorococcides is a species or responsible for anaerobic bioremediation of toxic trichloroethanol to non-toxic ethanol. So here the trichloroethane to non-toxic ethane. So this is being removed. So this Dechlorococcides is a species. They are responsible for the bioremediation of a toxic trichloroethane to non-toxic ethane. Okay, so that is being converted. And finally here we have the other one. It is called the Pseudopteopsis microspora. So this Pseudopteopsis microspora is a species of, one mark question, endophytic fungus, and capable of, this is all one mark question, which one is an endophytic fungus, capable of breaking down and digesting polyurethane. And this makes the fungus a potential candidate for bioremediation projects involving large quantity of plastics. Okay, right. So that's all about today's class. So this diagram part and all we will just discuss during our revision time. So diagram part, you just see the diagram and draw and get practice it. Nothing has been explained about this diagram part. Okay, so that is what I just wanted to tell you regarding this. So with this we are completing our chapter. So successfully we completed the second 25% syllabus. So 8th chapter is very, very important. So paragraph wise, topic wise you study. Every time one topic you make sure you are studying it. Okay, so that is important. So thank you very much.